Hi, this is Dr. A, and welcome back to your second lesson. This is on tools of the lab, and these are the tools that the microbiologist in the clinical lab uses to identify organisms that are causing disease and problems in patients. So, identifying the microbes that's causing a problem in your patient starts with specimen collection. So, specimen collection is actually one of the most important steps, because if you don't have a good specimen, then everything that you do following that is pretty much going to be worthless. It's we have a saying in the lab, it's called garbage in, garbage out. So if you don't have a good specimen, if it's not well collected, then what are you going to identify? Maybe there's not any organisms in there, maybe you'll miss the infection, maybe you'll misidentify it, or something like that. So some of the things that I do want to specify about specimen collecting is it's really important for you to note not only who it's from and to label it at bedside, but also to label where it comes from. So if you think about it in a couple of ways. So first of all, for example, this container that looks like a urine container, looks like it has some golden urine fluid here, and that's all well and good, but sometimes peritoneal fluid will look like that too, and so or paracentesis fluid or something like that. So it is very important to just not assume that a golden yellow liquid is urine, and that we actually properly identify. But if it's urine, it's also important to identify things like, was it voided urine, was it from a catheter, so there's things there. And then if you think about taking a swab and swabbing patients, you can swab up the nose, you can swab the back of the throat, you can swab a wound, you can swab all kinds of stuff. So just sending a swab down with a patient's identification without saying where it's from, again, can be very misleading and cause problems, because in the lab, we get swabs for rabbit testing, so like for viral testing, like the flu, or for the strep and stuff like that, but also for wounds. And you can't just assume that if you have a swab and you have an order that it's the right swab to the right order. So other than that, the steps after specimen collection include what we call the 5 I's of microbiology. So you do need to know that. The first thing is once you have a specimen, is you need to inoculate that specimen into or onto media that will allow the microorganisms that we're trying to identify to grow. Okay, so we're going to take an example of, let's say we have a urine, there's an obvious UTI that's bacteria in the urine, and we want to identify the bacteria. So the first thing we need to do is get some of the urine, and we're going to plate it, inoculate a media plate like this, and that will allow the bacteria to grow, because the media plate has everything that the bacteria needs to grow. Okay, so then the next step is going to be the I of incubation, and again, I'll show you in detail here in just a second. That allows time for the bacteria to grow in the right environment, etc. And then we're going to have isolation. So if you treat your plate correctly, you should have some isolated colonies popping up. And if there are multiple organisms in that urine, you should be able to see distinct colonies and isolate one organism from another. So maybe instead of just having a simple, let's say, a urinary tract infection with E. coli, they have a mix of E. coli and maybe staph or Pseudomonas or something, I don't know, Klebsiella, Proteus, who knows, right? So you want to be able to isolate and differentiate different bacteria. Inspection is noting what the bacteria looks like on the plate and stuff like that, and what plate, what type of media it grew on. But also we can then gram stain the colonies, and then staining, we're going to look at that, allows us to make some differentiation on what the bacteria actually looks like. So that would be part of the inspection, and then as you do the inspection, you can start to identify what type of bacteria you have. So we're going to go through all of these in detail. So obviously first, specimen collection, swabs, sometimes fluids are drawn via syringe, in a sterile cup, all kinds of different specimens. Again, make sure you label indicate what type of specimen it is, where it comes from, time of collection, data collection, and all of the, as much information as you can put on there that's pertinent is super important. So then we are going to inoculate. So typically the two most widely done methods of incubations are going to be the streak plate, as illustrated here, for the majority of your cultures, and then blood culture bottles are going to be it. Now, you can inoculate a bird embryo for viral cultures, but we do not do viral cultures in the clinical lab. It is more something that's done for research. Either way, once you have inoculated your media with the cultures from your specimen, then you will put them in an incubator. The incubator usually replicates the body environment, so it'll be 37 Celsius. And you can also have modify the environment if you need more air, less air, and stuff like that. There are different things that you can do to basically replicate the conditions in the body so that the bacteria will grow. So let's talk again a little bit more about inoculation. So to culture means to grow microorganisms, and we are growing them for the purpose of identifying them, right? Media, or medium as a singular, is the nutrient for the growth of microbes. Media can be in a plate, like we've seen so far in a streak plate. Sometimes it can be in a slant, and it can be pretty solid to liquid. There's all kinds of different forms of media. We're going to cover that a little bit more in detail, and you have an assignment on media too. An inoculum is a small sample of microbes, so that's what you would get when you, either like when you roll your swab on your plates, or when you put your loop into your cup of urine and you're picking up some urine fluid with some bacteria. That would be an inoculum, and that is what you transfer to the streak plate. And then inoculation is the process of taking that inoculum and putting it onto a culture, onto media plates, petri dishes and stuff to culture the microbes. Again, your clinical specimens are obtained from body fluids like urine or peritoneal fluids and stuff like that, maybe even poop. Discharges, think of pus and wounds and all of that, things you cough up. Anatomical sites, you can have tissues and stuff like that, sent in for cultures and stuff, disease tissues, all of that, scrapings, etc. Okay. The steps in a streak plate. So first, when you want to streak a culture plate, a media plate, you will start by rubbing your loop back and forth in what we call the first quadrant. And you don't want to go all the way across. You just want to go a little bit, not even a quarter of the way down, like more like, I don't know, an eighth of the way down, maybe. There we go. Okay, so then what you take, this is going to be where you're going to see the most concentrated growth. This is your swab straight from your wound. It's the urine straight from the urine cup and all of that, just from the loop. Okay. Then you're going to take your plate, you're going to rotate it 45 degrees. And what you're going to do now is, with your loop that you sterilizes, you are going to go back and forth into your first quadrant of inoculation. And then you're going to make a second quadrant. So you're going to go back and forth. So you're going to pick up some from here, and then basically you're spreading it out. You're thinning it. Then you're going to rotate your plate again, and you're doing it like that in a third quadrant, and then you rotate again, and you're doing like that in the third quadrant. And you flame your loop in between. So basically what you're doing, you're diluting it, so that you thin out the bacteria so that you can get isolated colonies. So on this example, there's a mixed bacteria here. So this is the first quadrant where they started, right here. This is the second quadrant right there. This is the third, and then this is the fourth. And you can see in the fourth quadrant, you finally are starting to see some isolated colonies. So we have a red colony, we have a yellow ones here, and you have some white ones, okay? And that's what you want. You want to be able to then, because then we can pick this one and cell culture it or do some other stuff, some tests on it. We can do a gram stain on it and all of that. and identify it, and then we can repeat the process for that one and repeat the process for that one, and we can identify all 3 types of bacteria that were in this specimen. Okay, so if your inoculum, if it's too heavy and you don't spread it out, you could have your whole plate look like this, and then basically you have to start over and you lose 24 hours of work and stuff, because you do want to get it thinned out where you can have isolated colonies. There's no way, even though you can see the red ones and all that, there's no way you can pick them out without getting some of the others on there. Okay, so then incubation is really simple. Again, it's just a temperature controlled chamber to encourage the multiplication of microbes. We usually use temperatures anywhere from 20 to 40 Celsius, usually 37 degrees Celsius, which is body temperature, 20 is room temperature, and 40 is a little bit warm. There are a few organisms that like a little bit warmer. It's like a body with fever. There are a few that like that type of environment. The atmospheric gases such as oxygen and carbon dioxide can be required for the growth of certain microbes. So the majority of the microbes that we're going to have deal with oxygen just fine, and so when you put them in normal room temperature, oxygen but in 37 degree temperature, they do fine. They grow the way they're supposed to. But if you had, let's say you drew a blood culture and the bacteria was an anaerobe, and you plated it, you need to put it in a CO2 environment. And so there's a very easy way to do that. There's basically some Ziploc bags with a tablet that's in a pack, and you put your plate in a Ziploc bag and you put the pack in there and you activate it and you Ziploc it shut. And what the little tablet in there does is it converts all the oxygen to carbon dioxide, and then it creates a carbon dioxide rich environment for the bacteria to grow. So the whole point of incubation, again, is for the microbes to grow, to multiply, and to produce visible growth in the media. Okay. Then from there, we are going to go over isolation, inspection, and identification. So again, isolation, if you have a good streak plate, then you can get isolated colonies. And then you might need more of it, and so you can, at this point in time, you could subculture. So we had the plate with all the mixed organisms, the red, the yellow, and the white one. Well, you could take some of the red one and put it in, and subculture it into a liquid or liquid broth or a plate, and have just that organism, so you can get it to multiply, you can get plenty of colonies, and you have plenty to test. And you can do the same thing with the yellow organism and the white organism, and then you would have, so from one plate, you would have, if you did a liquid and a solid one, you would have 2 for the red, 2 for the yellow, 2 for the white, and you would have plenty to work with for further testing. Again, inspection usually involves staining, putting so picking a colony, putting it on the slide, and then staining that slide so you can see what type of microorganism it is. You will not be able to see a virus. This is strictly for bacteria or fungi. So you can also note which media plate it's growing on, what color the colonies are, how the colonies look, and all that kind of stuff. So you can make all these observations via your inspection of the plates and the organisms and stuff like that on slides. So, and as you inspect and you get your microscopic morphology with the stains and stuff like that, that starts giving you the clues for the identification. The other ones is there are biochemical tests that can be done to identify the bacteria. There are immunological tests that can be done to identify bacteria, and then there are genetic tests that can be done to identify the bacteria. But first, you have to have isolated colonies so that you have the specific bacteria that were in that culture, and you have them in colonies. You have something to work with to run these other tests. Okay, so again, isolation is again based on the concept that if an individual bacterial cell is separated from the others on a nutrient surface, it will produce a discrete mound of cell called a colony. So again, here, each little mound here is a colony. Some of these kind of running together or have odd shapes. Some of them have nice round shapes. And again, that can be indicative of the type of bacteria that it is. So a colony is a microscopic. You can see it with the naked eye on the plate. Okay, it is visible on the plate and appears on a solid medium arising from the multiplication of a single cell into obviously millions of cells, enough that you can see them with the naked eye all in one cluster. So isolation requires a medium with a firm surface, such as a petri dish. Well, the petri dish that holds the medium, the agar is the medium, if you will, and an inoculating loop to transfer the specimen, and then to also streak the specimen on the plate. So again, the same three organisms. When you do you isolate, if you've done it correctly on the plate and you've subcultured correctly, then you should have pure cultures. So this is a slant plate, so you can see the white one is in here pure, the yellow one is in here pure, and the red one, and there's no mixing them up, okay? This would be a mixed culture because you can see there is an abundance of the white and the yellow, and they're all kind of mixed up. So this is a mixed culture. So they could have been potentially from the original culture where the patient got an infection with multiple bacteria, or it could have been a subculture that failed where you didn't pick a good isolated colony, or you didn't flame your loop good or whatever, and it was cross-contaminated with another one, and then you have a mixed culture here. Again, as long as you have isolated colonies, you have something to work with where you could subculture to yellow and subculture to white and then identify each one. And then a contaminated culture looks different from a mixed culture in that the one contaminant is usually single and in one spot and just in one quadrant and stuff like that. So they could come from maybe you coughed or sneezed or something landed on the plate. Obviously, before when a microbiologist is going to streak a plate, if you look at the plate, you want to make sure there's nothing on it to begin with. You would not streak a contaminated plate, but it is possible to get contaminants in there occasionally. The difference between this one and that one is that, again, this one is just one colony. It's just in one location. You don't see it all over the place, whereas this one you can see the white and the yellow are scattered all over the media. So this is a mixed culture. Again, this is illustrated so how if you had a mixture of bacteria, as you streaked it, you can spread these bacteria out where the parent cell of the white and parent cell of the yellow, or the cocci and the rod here, are separated, and they each can make their own little mound, their own little colony, and then you can see them. You can see hopefully you've streaked it correctly where the mounds are separate, and you can actually identify each one and visualize them differently. So there are several ways to achieve isolation. So I explained to you the streak plate right here, which is the most common method. And if you send a culture to the lab today, this is what they're going to set up. They're going to set up a streak plate. Occasionally, what you can do is you could set up dilutions. So to set up dilutions, you need sterile tubes here. And what you do is you put some of your from your specimen, you put it in this first one. It's going to be really concentrated, and then you take some of it and one loop of it and put it in this tube. So this is going to be more dilute, and then you get one loop of it and put it in here. It's going to be more dilute, and then you pour each one of those on a separate plate and you let them grow. And the one that's appropriately diluted will give you some isolated colonies. Now, this is a valid way to do it, but now think about this in terms of cost. With the streak plate, you are using one loop that you flame. Even if you use a sterile loop that you replace, you're using a loop that you can sterilize, and you are using one petri dish with agar. Okay, this one you still need just one loop. You need at least three, if not more, of these glass tubes, plus 3 different petri dishes with agar. So you need 3 times the supplies that you need in here, making it more costly, obviously, to do this technique. And then there's another one too. It's called the hockey stick one, where you just put some in the middle, and then you use hockey stick to just spread it out, and you would have growth throughout. If depending on the concentration of the colony, you may or may not get isolated colonies there. Concentration of the specimen, you may or may not get isolated colonies. But if you needed to have bacteria spread all over the plate for another reason, and you knew you're starting with a pure colony, this would be an appropriate way to get them to just grow all over the plate. Okay, so inspection and identification. So again, as you are inspecting the bacteria via staining and other methods, then you also are going to start running different identification tests, and you're going to be able to start coming up with an identification method. So microbes can be identified through their microscopic appearance. So this is the appearance under the microscope. So, for example, these are bacteria in a gram stain. Right here, we have some purple ones and some pink ones. Or here are some, these are malaria, right? Protozoans right here. So these are the eukaryotic organisms. They're parasites. And this is on a blood smear. Okay. And there is also, you can characterize the cellular metabolism. I'm going to show you, again, a strip that does that. So the idea of characterization of cellular metabolism is identifying a bacteria by what it eats, what it can metabolize. Some can ferment sugars, some can't. Some eat certain amino acids or certain sugars, but not others. It's kind of like you identifying who made a mess in the kitchen if you have a bunch of kids by like what was left behind, knowing what your kids' preferences are, kind of like that. So, nutrient requirements, the products given off during growth, presence of different enzymes and all of that, from which they derive energy can help you identify the type of organism that it is. And then, of course, where more and more of bacterial identification is going now is genetic and immunological characteristics. So your rapid strep test is an immunologic test, so it uses antibodies and antigen reactions. And you can actually do it directly on the from the swab, directly from the specimen without having to grow it out. But there are other ways too to use cultures that were grown out on a media plate on a petri dish, and then use immunological tests. And then to genetic testing, once you have isolated colonies, there are ways to identify the DNA sequence of the microbe, and then match that DNA sequence to a database. That will be something you're going to do virtually in your last lab. Okay, so let's start with our first open-ended question. Okay, so this one, obviously we're not getting in groups, but this is my traditional lecture. So what you're going to do here is write down, from the scenario, write down actions for each of the steps in identifying microbes that we just discussed. So a patient presents to the doctor with severe cough, fever, and sore throat. Okay, so what steps would you do for specimen collection? What would you do for inoculation? What would you do for incubation, isolation, inspection, and identification? So just write me the steps you're trying to identify the microbes from a patient that has a severe cough, fever, and sore throat. There you go. So just put what you think you would do there for each of the steps. Okay, so let's talk a little bit about media. You have a lab on media to help you like dive a little deeper into this. So the idea of media is providing nutrients in the lab so that the bacteria can grow and form colonies. The media can be found contained in a test tube, like here, a flask, which is bigger than a test tube, and petri dishes. So the test tubes and the petri dishes are what is the most used in the clinical lab. Media can be inoculated with an inoculating loop, like here. This is a plastic one that's sterile. It's a one use and done. The metal ones, you can flame through a loop, and you can use them over. You can flame the loop in fire, and then you can use them over and over again. The other end is called a needle. So you can do that to like stab some cultures into a tube. Pipettes, the pipettes have to be sterile, so they would be in a wrapping where you'd open and they're completely sterile, and then you can use it to get a certain amount of liquid and transfer it. And then swabs, obviously, so some of them have the proper way to transport it without contaminating, but basically big old cotton swabs. They should come in from a sterile package also. So again, sterile technique is necessary, so you don't want to be picking up bacteria and stuff from the environment, from your hands and all of that. So your test tubes, your petri dishes, your agar, all of that has to be free of any contaminations, has to be sterile. Your loop, your pipettes, swabs, all of those have to be sterile. Otherwise, you're introducing unknowns into the process that could completely deroute your whole identification process. So media can be found in different physical states. So they can be liquid. Usually if they're liquid, they're contained in a test tube with a lid. And there's think of a broth or something like that. They can be semi-solid. So semi-solid is more like the consistency of like hair gel. It's got some structure to it, but it's you can still kind of get it to flow back and forth and stuff. And then solid that can be converted to liquid, and solid that cannot be liquefied. Most media is made as liquid, boiled, and then poured into the plates. Although, and we can do this in the lab, although nowadays, obviously, most media are bought already the plates already made, poured and stuff like that, and wrapped in sterile containers. And so, they can be on a petri dish, they can be in a test tube. You can have solid on a slant in a test tube, so where you can go. And you're going to have solid in the test tube where you would poke it in. There's really quite a variety of format. So the media is usually agar. And agar is a complex polysaccharide isolated from gelitum, type of algae. It is solid at room temperature, but liquefies at 100 Celsius, which is boiling, and solidifies at 42 Celsius. Okay, so as it cools, it solidifies. It is flexible and moldable, so it'll take the shape of whatever you pour it in. And it is not digestible for most microorganisms, which means the microorganism can grow on top of it without breaking it down and liquefying it or something like that. Okay, so now let's talk about what is in the media. So what it is we have to provide the right nutrients for that bacteria to grow, but then sometimes, especially if you have a mixed colony, you may want to grow some and exclude others. Think about, for example, culturing stools, culturing poop. Poop is like nothing but a whole bunch of bacteria and waste material, right? And maybe you're just looking for the presence of some bacteria like Salmonella or Shigella or something that would cause a problem, but you're not interested in all the other guys that are supposed to be there in the first place. So there's ways to select the right media where you're actually blocking the growth of everything else and only encouraging the growth of Salmonella and Shigella if they're present, for example. So a defined or synthetic media is composed of precisely chemically defined components. They contain pure organic and inorganic compounds that vary little from one source to another. And so it would have a specific sugar or a specific amino acid, and there's a specific recipe that you can follow, and as long as you can obtain the single components, you can mix it in the right quantities and create that media. So it's chemically defined, okay? Very easy to reproduce. In complex, one of the components is not, chemically defined. And so, and they contain extracts of animals, plants, or yeast. So, and those things are complex. So if you think of all the components of blood, serum, meat extracts, or infusions, there is a whole bunch, meat infusions is what it means. There's a whole bunch of different types of amino acids and fats and carbohydrates and all of that. And so, and those are a little bit harder to replicate. And so those are complex, but we use them. You just can't go to your cupboard of single components like amino acids and sugars and stuff like that and grab them and mix them up and make blood media. It just doesn't work that way. You have to get blood from an animal. Okay, and so often we use sheep blood. General purpose media can grow a broad spectrum of microbes, quite a big one as possible, and it's generally complex. So sheep blood agar plate is a general purpose media that is complex because it's complex. It has so many different nutrients. It can actually meet the needs of a variety of microbes. And so this is a good one that we use. We often plate on blood agar media because it allows quite a few things to grow, and you can see what all is on there. Okay. An enriched media contains complex organic substances such as blood, serum, and hemoglobin, but also special growth factors, or they can also have special growth factors for the growth of what we call fastidious microbes, so microbes that need basically a special diet. They're picky eaters. Chocolate agar is a type of enriched media. It's made from on the basis of blood. Selective media are really interesting. So it contains one or more agents that inhibit the growth of a certain microbe or microbes. So for example, a Conkey agar plate is selective because it allows gram-negative to grow, but not gram-positives, for example. So that would be selective. You can also have plates that have, like I remember in the lab we had some MRSA plates, so there are plates to have certain antibiotics that will block the growth of all organisms except for MRSA. And then differential media, and a lot of media are selective and differential, both. It allows multiple types of organisms to grow, but to display visible differences in how they grow. So they can vary in colony size and color. Usually either the colony changes color depending on a specific characteristic, or the media changes color where that specific bacteria colony grows. So those are differential. So for example, if the basic plate is pink, but specific bacteria grow, it can turn that area of the plate yellow, for example. Or on a Conkey, if it ferments lactose, it will have a dark pink color, the colony will, but the media stays the same. Anyway, so a medium can also be selective, can be both selective and differential, and a lot of them are. So again, Conkey agar suppresses the growth of some organisms, especially the gram-positive ones, while producing visual distinction between the ones that do grow. So you have gram-negatives. If they ferment lactose, they're pink. If they don't ferment lactose, they're usually kind of a clear or white color. And often dyes are used as differential agents because many are pH indicators, and they change in color in response to the production of an acid or a base by the bacteria, which causes a color change in the media. So again, here's a mixed sample. On a general purpose non-selective, everything grows, but on a selective medium, only one species, one type of bacteria grows. So you can tell, and oftentimes we plate multiple plates, so we get a sample and we get a general purpose, and then we get some selective and differential ones. So we'll plate multiple different plates, and then you can see what's going on when you look at all of them together. And if it's a differential, so you can see the media has a certain color, the colonies have different colors depending on what they are, and you can even see where the media can change color. Okay, so at this point in time, we normally do an assigned media assignment. This has been done into a lab view, so you will be working on these. So I'm going to introduce some of these that we normally look at in class. So here's blood agar. So colonies can have various look on blood agar. It is made from blood, so sheep blood, so it's complex, and it allows for a lot of different types of bacteria to grow. So I do want to note, so if they make nice, pretty round colonies, they're considered non-modal. They don't move around. They just multiply and just mound up, mound up, mound up, mound up. Okay. If they're doing something like this, where it's irregular, it's more concentrated in the middle, or like this here, where you almost see waves, these guys are modal, and so they're spreading out from each other because they can move around on the plate. They can have different colors, from golden yellow to grayish to white. Really sharp white. They can look chalky or shiny, glossy and stuff like that, colors, depending on what type of bacteria it is. Another thing that happens with blood agar that you need to know is some bacteria can actually break down the blood cells in blood agar. So if a bacteria can actually completely eat and break down all of the cells to the point where the media turns from red to clear, it is said to do beta hemolysis. For that, then blood agar can be differential. Okay, if it breaks it down partially, so where it's leaving some of the residue behind, and it has more of a yellowish greenish tint to it instead of the media being completely clear, it is called alpha hemolysis. And the way you do it is you take the plate with the lid and you just kind of look up and have a light to shine through it, and you can really see if it's clear or if it's like greenish color, gray, not really gray, but kind of a greenish color. Or if it doesn't, if it cannot break down the red cells in the media, then it is called gamma hemolysis, which means no hemolysis. Okay, so chocolate agar is for fastidious, so hard to grow organisms. So it's an enriched type of media. You have, in the chocolate agar, you have also Thayer-Martin, which is a chocolate agar with a specific antibiotics and stuff, specific components that basically only allow Neisseria gonorrhea to grow. So if you use a Thayer-Martin plate, and usually what would we put on a Thayer-Martin plate is going to be a vaginal or penile swab, and you're looking for gonorrhea. If something grows on it, it's gonorrhea. If nothing grows on it, they just don't have gonorrhea, and that's just the way that one works. All right, Conkey. So this is the one that's both selective and differential. Okay, so the base is kind of a light pink in the Conkey agar, but if you have a lactose fermenting colony, it turns it dark pink, the colonies. If it is non-lactose fermenting colony, it stays kind of a whitish kind of color. In these, both Conkey allows only for the growth of like gram-negatives, enterics and stuff like that. So your E. coli can grow on here really well. And so here you get E. coli and I believe this is Salmonella here. One's a lactose fermenter, the other one is not. So you can see the difference in the colonies. So this one would be a Conkey with some lactose fermenters here growing and turn the colonies pink. Okay, so the Mannitol salt agar, this one is originally, when you set it up, the agar is pink. There's a pH indicator that changes to yellow if they can break down the mannitol, and they have to be able to grow in the presence of salt. So an organism that can grow with salt is staph. Staph likes salt. So if it's Staph aureus, then it can break down the mannitol and it will turn it yellow. If it's Staph epidermidis, it can grow on it, but it will leave it pink. Okay, so Staph epi is normal on your skin, so that's why it's epidermidis. So it wouldn't cause infections. It usually doesn't cause infections. It's an opportunistic one. But Staph aureus can cause boils and sores and stuff like that. So that would be one way you can tell the difference between the two. And then you have the urea broth and carbohydrate fermentation. So it's the urease test. So you put the bacteria in here. This is a slant. This is a broth. But if the bacteria does not use urease, this will be negative. If it does, it's positive. There you go. It turns pink. So yellow or pink or kind of orangey or pink. And these, so the initial broth is red. If they do not ferment carbohydrates, it stays red. If they ferment carbohydrates, which means they can break carbohydrates down, it turns yellow, so they can eat carbs. It's yellow. And if they produce gas in the process, you're going to have a gas bubble here that pops up. Okay, Salmonella Shigella agar. So this one is one that you would use to set up poop. And on a Salmonella Shigella agar, the Salmonella is going to turn this dark black colonies and all that. And Shigella has more this kind of grayish appearance there. So Salmonella Shigella, again, to isolate these two pathogens from stool where there's a whole bunch of bacteria that grows. Lowenstein-Jensen agar and Saburo. So Lowenstein-Jensen is this green agar. This is used strictly to incubate tuberculosis, TB, which is very slow growing. The cultures take about 30 days. But this is what it would look like on the Lowenstein-Jensen. And TB, if it's there and it grows on it. And then Saburos is good to grow fungus and yeast and stuff like that. So Candida albicans on Saburos and Trichophyton terrestre on Saburo. That shows agars. These are both fungi, not bacteria. So next, let's talk a little bit about preparing specimens for the microscope. I wish we could do this in person and stuff, but unfortunately we won't be able to. So there are several ways that you can put specimens on a slide for observation on the microscope. And so depends on the type of specimen you want to look at. If it's a living specimen or a dead specimen, that makes a difference. So a living specimen, what you want to do is put a wet mount. An example of that would be when we look at urine under the microscope. We concentrate the urine first, take out some of the sediment, pour off most of it, keep the sediment, put a drop of the sediment on the slide, put a cover slip on it, and then put it under the microscope. And you can see white cells moving around and stuff, but if there is Trichomonas, which is a little parasite that has a little flagella and it swims, if it's in there, you'll see it swim across your field on your microscope. And so you have to be able to see that motion and all of that to call that Trichomonas. And so that would be a wet mount. You wouldn't want to kill the specimen to do that. A lot of times when we look at bacteria and stuff, what we'll do is we will let it dry and heat fix it, and that essentially kills the bacteria. And so there's no way you're seeing any kind of motility at all on a dead specimen that's been dried and flamed and all of that. So again, what are the aims? What do you need to accomplish? Do you need to see motility or not, or whatever? And also, what kind of microscopes do you have available? We only pretty much have the bright field in the science lab. So let's talk about the fresh living preparations. There are two ways to do those. So they're either wet mounts or hang and drop mounts. And so again, to see them naturally, see them moving and all of that. Sometimes you can put a drop of stain to give some contrast, but again, the whole thing is going to be wet and moving around. Okay, so a wet mount is just a slide, a drop of the specimen, and a cover slip on top of it. And if you're not careful, the cover slip can slide back and forth. Hang and drop one, the slide has a hole, has a dip. You take the cover slip, you put the specimen on the cover slip, a drop on the cover slip, and then you hang that upside down and you drop it on the slide where the drop is hanging into the hole in the slide, indentation in the slide. And then you can often seal it and stuff. This one takes specialized slides that are more expensive. So guess which one do we do in the clinical lab the most? Ta-da, the wet mount. Simple slides, cover slips, very easy to do. Okay, so we can also use stains to better visualize the specimen on a microscope. So Unstained are harder to see, although you can if you use contrast and all that, but it is a little bit harder to distinguish things. So staining always makes things more visible. And so staining is any procedure that applies colored chemicals, dyes to specimens. A basic dye has a positive charge, acidic dye has a negative charge, and depending on the composition of the cell wall, the cell wall of the organism will attract either an acidic or a basic dye. So for example, if your cell wall has a negative charge, it will attract a dye with a positive charge, because negative attracts positive, and vice versa for if your cell wall has a positive charge, it will attract an acidic dye, because acidic dyes are negative. And and often this says acidic dyes are repelled by cells, so they could just stain the background too, because most of the cells have a negative charge on their cell wall, so the basic dyes is what will bind to them. So again, the bacteria like the basic dyes. All right, so a positive versus a negative stain. So it's really simple. The positive stain stains the bacteria or the organism itself and does not stain the background, so the slide stays clear. A negative stain, your organism stays colorless and the background is what is stained. The most used on the negative staining is India ink, and you're looking for an organism called Cryptococcus. And you do that on looking for cryptococcal meningitis on a spinal fluid. And so you put a drop of your spinal fluid, a drop of India ink, and a cover slip, all that on the slide. And if the cryptococcus is there, you'll see a really dark background and then really bright spots in the middle of it that aren't bubbles, that are actually organisms. If there's no cryptococcus, all you have is one big old dark slide, just black. It's just black all over, and you're just not seeing anything. The majority, though, of our staining are going to be positive staining. So the cell or the organism is colored by the dye. Some of the dyes that are used are crystal violet, methylene blue, saffron, and malachite green. If it's a simple dye, you're only using one of those, and whatever's there is going to be stained that color. So let's say you use malachite green, everything's there that is a cellular structure that can take up the dye is going to be green. Okay. If it's a differential stain, you're going to use two different components, and we're going to learn the gram stain, which is the most used, which uses crystal violet and saffron. And depending on the composition of the cell wall, some of the cells will be violets, crystal violets will be purple, and some of them will be the color of saffron, which is a pinkish color. There are also spore stains that can reveal spores, and acid-fast stains that can reveal acid-fast bacteria, which are like TB type bacteria. There are a few special stains. You can do a capsule stain, flagella stain, spore stain, granule stain, nucleic acid stain, and all of that. Those are used some, but not widely. The most widely used in the lab are going to be the gram stain and the acid-fast stain. So here are simple stains. So this is crystal violet stain of E. coli. You see the little rods here all over scattered, and then just kind of some junk in the background. And this is Corynebacterium with methylene blue right there to have varying shapes. This one. These are differential stains, so you have a gram stain. So you have these gram-positive cocci that stain a really dark purple, almost black, and then you have some gram-negative rods that are pink here in the background. The acid-fast stain, the reddish-purple stain cells in bacteria are acid-fast, so they hold the acidic dye, and the blue cells are not acid-fast. So if you saw some reddish bacteria like that, then you would assume that it's an positive acid-fast bacilli, could be a TB. And then the endospore stain can reveal spores versus vegetative cells. The endospore stain red, and the vegetative cells, which are actively dividing, stain blue in this one. A bit harder to see. The India ink one here. So I know this looks more like gray, but this is what Cryptococcus would look like on India ink. So you can see the organism and its wall around it. And so you see these basically little round organisms with halos around them. And if you see that, that is definitely positive for Cryptococcus. If it was negative, the whole field looks like this dark kind of color right there. Flagellar stain, again, not done a lot, but it's one of the special stains used. Okay, this stain, and so for here, let me move my here. If you look at the picture here, and you can click on it to see it better, is it a negative stain or a positive stain? Okay. And then next we have, this is the same organism from the previous side. So is this a positive or negative stain? Okay. Next one, we'll look at it. So is this a simple stain or a differential stain? Again, you can click on the photo to see it better. And next one is just your last open-ended question. Do you have any questions? So again, as you work through your media lab and your gram stain lab, you're going to get a better understanding of how these guys, how this works, and you're going to also work on your microscope lab. Now, the gram stain lab is coming up next. While you're working on tools of the lab, you have labs on microscope and labs on the different types of media. Okay, so if you have any questions pertaining to this lesson or anything that you need, just pop it in here, and again, I'll answer it as an announcement. Again, thank you for your attention. I will talk to you soon.